Okay, so I want to thank everyone that showed up here and welcome you to our eighth um, episode, if you will, session for this semester of our chemistry seminar. Today, I'm very excited actually, because I get to meet someone who I think is worthy of the phrase role model. Not only is she a highly recognized scientist, mathematical biologist, but she is actively involved in mentoring the next generation of mathematicians and scientists, especially from historically underrepresented groups. And I'm really excited that she accepted our invitation to tell us about her work. You will hear more about that when our student, John Rosenberg, introduces her. So my role now is to introduce John. So John Rosenberg, he's currently actually in my organic chemistry lab class, and he's doing well in it, John. Asks lots of good questions, seems pretty eager. So very good student. He's a sophomore biochemistry major. And he's actually minoring in painting and drawing. That's a pretty nice stretch of talent there, John. And he did his high school is that Loudonville Christian School? Is that in New York, John? Yeah, you could unmute.
Yeah.
In New York, okay. And one of his career goals, this is interesting, he wants, as a career goal, a cheerful, fulfilled life. Don't we all, John? With whatever we do, we hope it's cheerful and fulfilled. And a fun fact about him is that he loves fall. Again, I'm pretty sure there are lots of us who would agree with John on that. So John, this is your turn to introduce our speaker for today.
Okay, thank you for the introduction, Dr. Murray. And so our speaker, Dr. Trachette L. Jackson, earned her PhD in applied mathematics from the University of Washington. And 10 years later, after she did her postdoc at the Institute for Mathematics and its Application at Duke University, she was promoted to full professor of mathematics at the University of Michigan. And in 2003, she became the second African-American woman to receive the prestigious Alfred P. Sloan Research Award in Mathematics. And in 2008, Diverse Magazine honored her as one of the year's emerging scholars. In 2010, she became the first woman to receive the Blackwell Tapia Prize, which recognizes a mathematician who has contributed significantly to research and has served as a role model for mathematical scientists and students from underrepresented minority groups. And Dr. Jackson's research lies within the overarching field of mathematical biology, and she specializes in computational cancer research or mathematical oncology. Today, we are honored to have Dr. Jackson talk about her research, which focuses on developing mathematical methods to address critical questions associated with tumor progression and targeted therapeutics. As it should also be noted that she has built her career on collaborative research and educational activities that cut across traditional disciplinary boundaries, and envisions that this type of research will eventually change the face of cancer research. We are very honored to have you talk about your research today, Dr. Jackson.
Thank you. Thank you so much to Dr. Murray for the invitation, and to John for that really wonderful introduction. I feel like you know a lot about me and how I got to where I am today. And in that introduction, you heard there was a lot of mathematical training that went into my career path. And so in today's talk, I'm going to try to share with you how someone with my background, which was mostly mathematical, came to use her skills to help turn cancer discoveries into effective treatment strategies. So how we use these quantitative approaches to aid the biomedical sciences and oncology in particular. But before we do that, I wanted to share with you this excerpt from a Time magazine article. So on the cover, you see they said, "How to Cure Cancer." And one of the taglines in the interior was that the hero scientist who defeats cancer will likely never exist. And then they went on to describe cancer in the way that only a Time magazine writer can, so colorfully. So they said cancer is an intricate and potentially lethal collaboration of genes gone awry, of growth inhibitors gone missing, of hormones and epigenomes changing, of rogue cells breaking free. It works as one great armed force, attacking by the equivalent of air, land, and sea in stealth. And we think we're going to take it out with what? A lab-coated sniper? So in the beginning of that description of cancer, they're really telling you why this is such a formidable disease. And at the end of that description, they make a little bit light of it, saying, what do we think we can do against something like this, right? And that question really hits home. What can we do? And are we doing the best we can do? And why won't there be this one hero? Well, the reason there won't be this one hero is probably because cancer is not one disease. It's the name given to hundreds of genetic diseases that present the way cancer does, right? So it's not going to be one hero, but there will be many heroes, right? And in the article, they talked about all of the complementary expertise that might need to come to the table to help fight cancer. Of course, medical oncologists, basic biological scientists, biomedical engineers are doing wonderful things, informaticists who are getting us a much better genetic picture of what's going on, all of these people coming together. Now, this article appeared, several years ago, and they did not include mathematical modelers. So you see that I included us right here. And I did that for a reason, because hopefully through the course of this talk, I will convince you that people with this type of background, this quantitative mathematical background, people who develop models of cancer and the way it works and the way treatment strategies work, deserve a seat at this table, because we have an interesting and important contribution to make to cancer research. And it's this team-based cross-disciplinary approach to cancer that is really going to upend tradition and hopefully help us to deliver results faster. So the example I want to give you today comes from a particular problem that I've been working on for a while, and that's targeting cancer stem cells. And I'm going to give you definitions for what all of that means and how we use mathematics to try to help tumors that are driven by cancer stem cells and therapies that actually target this very special cell population. So first, I wanted to let you know what kind of cancer my collaborators on this project are working on. They are working on head and neck cancers. And that's a whole group of cancers that present around the head and neck area. They're the 6th most common type of cancer worldwide, and 95% of them are of a particular type, and they are squamous cell carcinomas. So throughout the talk, I'm just going to abbreviate that as HNSCC. And so that's the kind of cancer we're talking about. Not only is it a common cancer, but it's a cancer that is sort of stalled in terms of what we're doing to combat it. The 5-year survival statistics haven't changed in over 3 decades, really. What we're doing to combat this particular kind of cancer is a type of chemotherapy. It's the mainstay for non-surgical treatment. It's the best we can do, a particular type of platinum-based chemotherapy, right? So the question becomes, what do you do when the hallmark of medical treatment for the type of cancer you have, which is a cytotoxic chemotherapy, what do you do when that fails? And it fails a lot, right? If it didn't, we would be in a much better situation with how we're treating cancer. Well, the answer might be targeted therapeutics. And when I say targeted therapeutics, I just mean drugs that are specifically designed to interfere with certain cells, enzymes, proteins, or other molecules that are necessary for a particular tumor's growth dynamics. So all tumors are a little bit different, and those targets might be different depending on cancer type. Primary goal of this type of targeted therapy is to fight cancer with a little bit more precision than chemotherapy does and fewer side effects than chemotherapy has. So that's the goal. So now we know what kind of therapies we want to look at. Let's look at the kinds of cells that we're talking about. I said we wanted to talk about cancer stem cells. I'm abbreviating that CSC. So you'll see that abbreviation throughout, cancer stem cells. They have been discovered in a wide variety of malignancies, and they are the drivers of tumor growth. They have several special qualities when they are present within a tumor. They're almost always the minority. They only make up a small fraction of the entire tumor mass. They have special capabilities. They're able to self-renew, just like a regular stem cell would. A non-cancerous stem cell is able to self-renew an unlimited number of times. So are cancer stem cells. They're able to differentiate into different cell types, just like normal stem cells can. And probably the most frustrating aspect of these cells is they are the ones that are the most difficult to treat. They are often resistant to the chemotherapy drugs that we try to deliver, can shield themselves from radiation and cell stress, other things that would normally trigger a cell to begin the cell death cascade. They protect themselves from these kinds of things. So even though they're a small population, they play a disproportionately large role in tumor growth dynamics. So what we want to do is try to build a simple mathematical model of cancer stem cell driven tumor growth. And when you're developing a mathematical model to try to describe a biological system, you first try to understand as much as you can about the underlying biology. It's going to be really complex. So you have to simplify that down just a little bit. So this schematic on the top shows the true situation of what happens in biology. We might have a cancer stem cell that can divide symmetrically, giving rise to 2 new stem cells, or divide asymmetrically, giving rise to 1 cancer stem cell and a progenitor cell. Those progenitor cells further differentiate along their path until they become fully differentiated, and those cells just stay around in the tissue and eventually die. So if we wanted to write down a mathematical equation for every single one of these cell division stages, that would be a very, very large model. So instead, we're going to simplify things. We're going to lump all the progenitor cells into one compartment. And so our simplified schematic diagram of the mathematical model we want to build considers cancer stem cells that can self-renew, creating more cancer stem cells, and that can differentiate, creating progenitor cells, and they can die. We lump all those progenitor cells together, as we said, into one compartment. They come from cancer stem cells, but they amplify themselves. So we take into consideration that they amplify, and then they're able to terminally differentiate into that final cell pool. So we've reduced this down to 3 cell populations that we're really interested in considering. So we've done the first step in mathematical modeling, taken a biological problem of interest, and we've simplified it down into something that's a little bit more manageable. Next thing to do is to go from this schematic diagram, this picture of what's going on, into a mathematical statement of what's going on. So we have to translate this picture into mathematical equations. So let's do that for each cell type. So I know I'm talking to a lot of chemistry, biochemistry majors, maybe some students from biology, maybe some students from math. I know most of you have probably seen calculus and the all-important derivative. So here we use derivatives to describe the rate of change of our population. So you'll always see on the left-hand side a derivative representing the rate of change, in this case of the stem cell population. And then we write down functional forms that represent the self-renewal possibility that this stem cell self-renews, and there's some simple possibility that the cell dies at some rate, right? So now we've got a mathematical expression for this first population. We're going to do the same thing for the second population, our progenitor cell population. If delta S is the death, the rate of stem cells, alpha S is the proliferation rate of stem cells, but then there's some probability that that division will result in symmetric division. And 1 minus that probability is the probability of asymmetric division. If it's an asymmetric division, I'll get a progenitor cell, which amplifies itself a number of times. It can differentiate at some rate and can die at some rate. Okay, so we've got 2 equations of the 3. Our last equation just represents what goes on with our fully differentiated cell type. When a progenitor cell differentiates, it differentiates into 2 terminally differentiated cells, and those cells just stick around in the body until their time to die at some rate, delta D. Okay, so we've done another step in the mathematical modeling process. We have now gone from a biological problem to a schematic diagram to mathematical equations. Now we want to extract some information from a set of equations like this. And so maybe the first biological question that we would ask ourselves is, will an initial number of cancer stem cells form a tumor, or will they die out? So if I start off with some number of cancer stem cells but don't have any other cell types, am I going to get a fully formed tumor made up of all these cell types? Or is that initial population just going to eventually go to 0 and the cancer cells actually be cleared? So that's the biological question you may want to ask first of a mathematical model like this. And it also corresponds to a very specific mathematical question. Right? So the mathematical question that we could ask is, what are the steady states and are they stable? So we asked biologically, let me go back, what happens eventually to this tumor? What happens in the long run? So mathematically speaking, we're looking at what happens as time goes to infinity, as we eventually to this population. What's the long-term outcome of this population? Some of you have probably heard of steady states and stability. But let me just, for those of you who haven't, let me just put it to you this way. When we say the term steady state, all we're looking for is a solution that no longer changes with time, right? So we're just looking for a constant solution, static situation that usually is approached at long times. To find these steady states, there's a pretty easy mathematical way to do it. We just set all of those time derivatives equal to 0. So here's our model, our system of model equations that we wrote down. We can set those derivatives equal to 0. And we start looking for solutions to this system of 3 equations. Well, very clearly, if you look here, if S equals 0, that whole thing 0, so we're good. S equals 0 works. If S equals 0 in this equation, that whole term goes to 0, implying that E has to equal 0. If E equals 0, we can cross off this term and we'll have D equals 0. So we've got our first steady state. Our first possibility for long-term behavior is a cancer-free steady state. No cells present at all. This would mean the tumor dies out, is eliminated, and the body is cancer-free. Okay, so remember our question was, is the tumor going to die out? Are we going to be cancer-free? Or is a tumor going to actually form and persist in the body? Well, our first stent at analysis tells us that there's a possibility that this tumor is just going to die out, which would be great for the body. But we don't know that for sure. We have to ask the question of, is that steady state stable? Am I actually going to approach that long-term solution, in the long term? So if we want to answer that mathematical question, we have to look at a concept called stability analysis. It just answers the question of what happens if you slightly perturb a solution like this 0, 0, 0 cancer-free steady state. What if you slightly perturb away from that? Will I return to that steady state, or am I going to keep moving away from that steady state? If I return to it after a small perturbation, we call it stable. If I don't return to it, if I move away from it, we call it unstable. So just as a little pictorial analogy of that, I've sort of drawn very badly this hill valley representation here of this curve, and I've labeled points 1 through 4. Some of these positions can be thought of as steady states, because if I rest this ball, for example, right at the right spot here, it won't move, right? So solutions will not change with time. And if I do the same thing with this position, if I balance it just perfectly, solutions won't change with time. It should just sit there. And the same thing here. But if I were to put the ball here, it's gonna slide on down. So that would not be a steady state. Now, let's see if we can use the same picture to figure out some aspects of stability, right? So stability says if I wiggle the ball, am I eventually going to return back to that same steady state or move away from it? So if I wiggle the ball that's in position 1 or in position 4, clearly I might have some transient movement, but it should eventually settle back down to that steady state position, right? But if I wiggle the ball at position 3, even slightly, it will probably roll down the hill one way or the other way, meaning that steady state is not stable. So this is the idea we want to apply to our cancer-free equilibrium. Am I going to approach a state where the tumor is eliminated, or am I going to approach a state where the tumor moves away from that elimination possibility, the possibility of elimination? All right, so we have our cancer-free steady state. We understand a bit about this concept of am I going to approach this steady state or move away from the steady state? Got to do a little bit of math to answer that, but it turns out there are 3 quantities that determine whether this steady state is stable and I actually approach it. For those of you with a little more math, you might recognize these as eigenvalues. And we want to make sure that they're negative. We just want to make sure all 3 quantities are less than 0. So let's start at the bottom. The third quantity, lambda 3 here, clearly always negative if the death rate is positive, which it is. Lambda 2. Again, always negative, don't need to worry about that. Our parameters, our rates are always positive, so we're good. But then if we move up to the top here, our first lambda value, we see that there's definitely a possibility for this to be positive. If it is positive, our steady state is unstable and we will not clear the tumor. If it's negative, our cancer-free steady state is stable. So then we can write down the condition for stability pretty easily, right? The cancer-free steady state is going to be stable as long as the death rate of cancer stem cells, delta S, is large enough. And large enough being greater than the maximum proliferation rate, the maximum self-renewal rate. Okay, intuitively speaking, you probably could have guessed that, right? If I'm dying more than I'm renewing myself, right, I should eventually just die out. If I start with the number of cells who are dying faster than they can renew, then they probably won't survive. Okay, so mathematically, we get a result that intuitively makes sense. So here's our answer just again to our first biological question. If the death rate is greater than the maximum growth rate, an initial number of cancer stem cells will just die out. So let's ask another question. And that question is, what if the death rate is less than the growth rate? What can happen in that case, right? Yeah, sorry. There's the new question. What if the death rate is less than the maximum growth rate? What will happen in this case? Now our steady state is unstable. Our tumor-free steady state is unstable. Will it move towards a new steady state? And so we did a little bit of looking at what happens when our derivatives were 0. But now if we remove the possibility of all those 0 steady states, we can do a little bit of algebra to find a non-zero steady state. This steady state represents the tumor persisting in the population in your body. So the tumor is present at some constant level all the time. Okay. And if this steady state is stable, that means the cancer will survive in your body and will not be eliminated. So here's the mathematical description of what that steady state is. And if you do that same kind of stability analysis, you get 2 conditions this time that tell you whether this steady state is stable. So in this case, we have to have the death rate less than the growth rate, which we said, that makes sense. The cells are not dying faster than they're growing. But then there's also this other bound on the death rate. It has to be big enough. If it can't be too small, right? It's got to be small, but not too small. It's got to be bigger than the minimum possibility for self-renewal. So we kind of get these two conditions for this steady state to be stable. If the steady state is stable, we're going to approach it. It's going to be a nice constant solution. So let's look graphically at the two possibilities. Condition 1 and 2 hold, and condition 1 and 2 violated. So if condition 1 and 2 hold, we would get this blue dotted line. The tumor is just going to approach some steady state. This is tumor volume versus time, and the tumor is just going to approach some steady state. All right. But if that second condition is violated and the death rate is too small, right now I'm dying even less. My cells are staying around even longer. I go from a bounded situation of bounded number of cancer cells to unbounded growth that's going to just increase forever, right? So that's the worst case scenario. As I keep decreasing this death rate, I keep getting worse and worse situations. So a high death rate, tumor is cleared. A medium death rate, I get a constant level of tumor cells remaining in the body. Death rate even smaller, I get this tumor shooting off to infinity. Okay, so our mathematical model so far has predicted 3 types of dynamical behavior: clearance, constant, exponential, right? So you might be wondering, okay, so this has all just been math. Can really find situations where actual tumors grow in these different dynamic ways. So now what I'm showing you is data from experimental data for fit to this mathematical model for different situations. So this first set of data is from a primary human tumor from a patient who has squamous cell carcinoma from the head and neck. So that means they took the cancer cells from a patient, put them in a mouse, and let them grow. And in this case, when we fit the model to the data, we get this nice, very nice fit. The data points are the circles, and the mathematical model is the solid line. The data only went out to 120 days for ethical reasons with the mouse. But if you had let this grow out, you would have gotten that nice constant tumor growth here. Now, what if we look at a cell line's growth? So this is a human tumor cell line that had been immortalized. It's still a squamous cell carcinoma from the head and neck, but it's an immortalized cell line. And now in this case, we definitely get a very different growth dynamic. The data shows a different growth dynamic. The dots, our mathematical model fit shows that growth dynamic of exponential growth. And in this case, as we said, the death rate of these cells turns out to be much smaller than the threshold necessary to keep this cancer under control. These cells are living too long. So far we've shown the model can predict things that actually happen with the experimental data for tumor growth. Let's just show you one more thing the model can do. You might want to know what the tumor is made up of. We have 3 different cell types. So what cell types are dominating as this tumor is growing? So let's break this down on the columns. The first column looks at tumor size, and then the rest of the graphs, the other 4 graphs, look at tumor composition. In the first row, we're varying that delta S, that stem cell death rate, which was also important. That's the one that if it got too small, we could get exponential growth, right? So first of all, just looking at tumor volume versus time, as the death rate gets bigger and bigger and bigger, the tumor size gets smaller and smaller and smaller, as we would expect, okay? But now let's look at what the tumor is made up of for the largest delta death value and for the smallest value of the death rate. So for the smallest value of the death rate, we see that about 90% of the tumor is made up of differentiated cells. Very small percentages are made up of stem cells and progenitor cells. For the largest death rate, The blue curve, again, is differentiated cells. They make up a smaller fraction, about 60%. And look what happened to the stem cell fraction. It bumped up to around 30% of the tumor was made up of stem cells if we just increase that death rate. Just to contrast that, if we look at varying the progenitor cell differentiation rate, we can actually see progenitor cells take over this tumor, right? So for the smallest value of differentiation, we see the green curve, which are the progenitor cells, making up the dominant proportion of the tumor. So that means that the differentiation rate is so small that the progenitor cells are staying around a long time and they end up dominating. If we increase that parameter and let them differentiate faster, of course, we get fewer of them, and the differentiated pool again takes over as the dominating cell type in the tumor. So the model can actually predict what cellular composition your tumor will have as well. Okay, so we've done a lot without treatment. The whole goal, and even the title of the talk, had to do with treatment. How can we use modeling to impact therapy? But before we can impact therapy, we have to make sure the model works for tumors that grow without therapy. But it's time to add in a therapeutic approach. So conventional approaches like chemotherapy, which I said was the mainstay for this kind of cancer, what does it do? So if the blue cells are the normal cancer cells and the yellow cells are the cancer stem cells, which make up a tiny fraction, if you give chemotherapy, it's going to kill all the blue cells because these are the stem cells are resistant. They're not dividing as fast, all of those reasons, right? So it's going to look like when you go back and measure your tumor volume, it's shrunk a lot. It looks great. But if you wait, that cancer can always recur because you left that driver cell, the cancer stem cell, behind. So what about an approach that does the opposite? What if instead of concerning myself with the bulk of the tumor, I focus on targeting this driver cancer stem cell? I give a therapy. I go back and measure my tumor volume. It looks like it didn't work. The volume of the tumor is still almost the same because I've only killed off a very small portion of the cancer cells, right? But if I come back later yet again, if I've really cleared all of those cancer stem cells, all of these cells will have fully differentiated and will eventually die out. So that's a novel, non-conventional approach, is targeting these cancer stem cells. Let's see if we can figure out how. So again, the title of the talk was Turning Cancer Discoveries. So the biological sciences and clinicians who kind of work on cancer make all kinds of discoveries. And one of the things they discovered was that these cancer stem cells actually live near blood vessels. So here's an actual picture. Here's the blood vessel. You see it branching off. And then cancer stem cells are kind of stained in red. They kind of try to stay close to the blood vessel. And this is just a cartoon of that same thing. The cancer stem cells try to stay as close as possible to the blood vessel because things that the blood vessel cells are producing, endothelial cells are producing chemicals that help them maintain their special properties. So that's the first discovery. The second discovery is a particular signal that these stem cells need that endothelial cells in the blood vessels are making. So cancer researchers discovered that these blood vessel cells are producing a particular chemical, IL-6, and that's what's contributing to these stem cells self-renewing more, staying alive longer, moving more, all kinds of things, all kinds of stimulatory effects on the stem cells. So now I think we have our target. If we can target the IL-6, we are targeting those stem cells and maybe reducing those special qualities that they have. Turns out there was already a drug out on the market that targeted IL-6. It was being used for arthritis. But maybe we can repurpose that drug to see if it really reduces cancer stem cells in the head and neck cancer situation. So what this data shows you is the percent of stem cells before treatment and after treatment, in vitro and in vivo. So this particular treatment is an antibody against IL-6. I'm going to call it TCZ. When you give TCZ, you definitely decrease the stem cell pool in vitro, and you decrease it even more in vivo. So it looks like this could be a potential way of targeting cancer stem cells. Okay, so now we are adding to our modeling goals. We know that the standard of care, which is a chemotherapy, cisplatin, I'm going to call it cis, and then our targeted therapy, TCZ, affect the cancer stem cell pool in opposite directions. If I just give chemotherapy, I'm going to end up with all stem cells. If I just give targeted therapy, I'll end up with all non-cancer stem cells, right? So we want to use mathematical modeling to propose how to give both of these drugs together. Let's combine these drugs and try to get the best possible outcome. And maybe a mathematical model can help to do that. So we saw that simple model that we kind of developed together. We walked through the developmental stages of it. We have to blow that model up, make it much bigger and much more complicated, but still retain those features that we had in the simple model. So we still have stem cells that become progenitor cells, that become differentiated cells. But now we also have these blood vessel cells here over here that produce signals that the stem cells like. All right, so we add all of these different features to our simple mathematical model in order to make therapeutic predictions. I just wanted to show this slide really quickly because you might be wondering, okay, what kind of data do you have to have in order to make sure your model is working correctly? And I just wanted to show you that we did have a lot of different forms of data to test and train and collaborate and to calibrate our mathematical model before we ever tried to start making therapeutic predictions. So we had pharmacokinetic data on how each of the drugs behaved. We had tumor volume versus time data for various situations for how the tumor would perform when under different circumstances that we could adapt the model to address parameters. So we had all of this data to help us train the model. Now, I just want to show you a couple of slides that show what the model predicts. So here's a model prediction of a comparison of the model to data for combination therapy. So let me orient you here. What you're seeing in black circles are what happens to the tumor when you don't treat. And then the solid line is what the mathematical model fit to that data would look like. So you see that the black line is pretty much going through all the black circles. And then in blue, what you're seeing is two different treatment scenarios. They started combination treatment on day 23 or day 28. And all what you're seeing with the blue lines, the dashed and solid, are mathematical model predictions of what happens if you follow those treatment protocols. So no additional parameter fitting, no fudge factors, just overlay the model's predictions to the data. And this was our validation step. Our model is working. Our model predicts trajectories that look pretty close, very close to what the data is predicting. So we're happy with our model and how it predicts the tumor volume to change under therapy. The graph on your right shows what happens to the stem cell pool, just making sure that if you don't give therapy, you might expect, here's what you would expect for the stem cell pool in black. And then if you give these two therapeutic approaches, that you definitely are decreasing the stem cell pool, which is what you want to do. So combination therapy counteracts the effect of chemotherapy, increasing the stem cell pool. So the model's doing pretty good, but we want to use this model. It's a tool. What do we want to use it for? We want to use it to optimize how we give these two drugs together. So this picture just showed they gave both drugs at the same time and started that process on 2 different days. Maybe there's a better way. Maybe we could actually reduce this tumor even more if we did a different type of dose scheduling strategy. So here's what we want to do. We want to minimize the amount of targeted therapy required for a fixed amount of chemotherapy. We phrase it this way because, as we said, chemotherapy is the mainstay, been giving it for 3 decades. They know exactly how much to give and when to give it, right? So now we're adding a new therapy onto it. We want to give the least amount of that new therapy and get the biggest bang for that buck. At the same time, we want to maximize the cancer stem cell reduction. We want to get the most reduction in the cancer stem cell pool as possible, and we want to end up with the smallest tumor we can. How can we do this? Well, maybe if we change when and how often we give these drugs with respect to each other, we can do better than just giving them both at the same time. Okay, so that's what we want to do. So here's our plan. We're going to look for scenarios where we give different dose scheduling regimens. Okay, so we're going to start with our targeted therapy. We're going to look at giving it for 3, 4, or 5 weeks. And then we're going to either pre-treat, co-treat, or post-treat with our chemotherapy. That's the cytotoxic drug, so we're not going to give it as long. We're either going to give it for 2 weeks or 3 weeks. Okay, so that's the administration schedules we're going to start looking at to see if we can do better. Now we need some metrics to understand, are we doing better? By what are we going to judge that? Well, we're going to look at a couple things. We're going to look at what we're calling the IC70, and that's just the amount of the targeted drug, the new drug required for 70% reduction in the tumor by week 6 or week 8. So we are looking for substantial reduction in 2 months, right? We want this tumor to be shrunk as much as 70% by then. It's one metric. Then we're also going to look at another metric to see how these drugs are working in concert with each other. So we're going to look at the ratio of the IC70 for each dose schedule, right? And so we're going to take we're going to compare that to the baseline. So the baseline is the co-treatment case. That's what they normally do. Let's just give both drugs together. That gives us a certain IC70. We want to take the ratio of that with any new drug schedule that we try. If we get a number that's bigger than 1, that means we're using more drug than we were using. And so things aren't working well together. There's no synergy there. But if we get a value less than 1, we're actually using less drug than we were using. And there may be some level of synergy going on between the 2 drugs. So we want to look for that. Finally, again, we want to make sure the whole goal of this was to reduce the stem cell pool. Are we actually doing that job? So we want to look at those 3 things to determine how well our new dose schedule, our proposed new dose schedules are working compared to the old dose schedule. All right, so I think the next slide is going to look a little jarbled a little bit, but I'm going to walk you through it. So I don't even want to show it yet till you're ready for it. Get ready. All right. You're ready. So let me tell you what all this means. So the numbers down the columns, the counting 1 through, I think you can probably see through 35, that's just the different trials we ran. So we actually ran way more than this. I'm showing you a snapshot of different trials that we ran, simulations that we ran for drug combinations. Everywhere you see the word cis means I gave cisplatin, the chemotherapy. So just think of this as chemotherapy. Everywhere you see a gray shaded region means I gave targeted therapy. Remember, I said I was going to give it for 3 weeks or 4 weeks or 5 weeks. And remember, for chemotherapy, we were going to either give it for 2 or 3 weeks. Okay. Now, we were going to also, we said we could pre-treat with chemotherapy. We could give it at the same time, or we could give it after. So we're testing all these combinations. I'm showing you just a snapshot of the numerical simulations we did of the model to test all of these combinations. And then in these columns over here, we have those 3 metrics we wanted to talk about, right? We wanted to know, are we significantly reducing this tumor by 70%? Is there synergy going on? Are we reducing the stem cell pool? So these are the dosing strategies. They're pretty conventional. Either give all 3 doses together before, or start at the same time, or start, stagger it and give some after. Everything's pretty conventional. But when you look down these rows, first of all, are we reducing the stem cell pool? Yeah, by half in only 2 months, right? Actually, this is after 6 weeks. So less than 2 months, we've reduced the stem cell pool by about half, no matter what treatment strategy we use. If we look at the IC70, this is a measure of the amount of drug used, and it's a substantial amount of drug being used. But the worst thing is the synergy. Remember, we said we wanted numbers less than 1. These drugs are not working well together at all using these conventional dosing strategies. Almost every scenario gives me a number bigger than 1. These drugs are working in opposition of each other. So just check the time, we decided to look at non-conventional dosing strategies. So again, here are some of the simulations we did. Now look what we're doing. We're spacing out. We're not giving all 3 chemotherapy treatments together. What if we space out? What if we give chemotherapy, then give targeted, then chemo, then targeted? Or what if we actually alternate these things, right? So we looked at these non-traditional treatment regimes, and we ran thousands of simulations of all these things. And now if we look down our synergy category, for the first time, we start seeing a number less than 1 right here, right? And that's when we started treating them both together, right? We gave the first dose of cisplatin, the chemotherapy and the targeted therapy together. And then we took a week off of the chemo. And then we took a week off and we gave it. And that's the first time that we saw a number less than 1. Now, if we still scroll down, we see we can get these drugs working really well together. We can get a number way less than 1. If we pre-treat, in this case, with the targeted therapy, followed immediately by chemo, and alternate that pattern, that's when we get the smallest synergy factor, meaning the best synergy result. And look how much drug we were giving. So much less drug is being given. Let me just remind you, these were all in like the high teens and 20s over here. Now we're giving way less drug and having them work better together. So the model says giving these drugs at the same time is not the best you can do. You can do much better. Use less drug and have them work well together and get huge reductions in 6 weeks if you do that. Okay, I want to stop here and just summarize really quickly. We developed this mathematical modeling framework. It operates at all these different levels. And the big thing we wanted to do with it was capture the mechanism of action of these targeted therapies against cancer stem cells. And then we wanted to use it to investigate the crosstalk between the tumor and the blood vessel, which is producing things that the tumor needs. And figure out how all of that interplays with the reduction we want to see to therapy. And then the best thing I think we did with this model was try to use it to predict optimal dose scheduling regimens and potential synergism or antagonism of these drug combinations. So that's what we've kind of done here. Just a picture of some of the people who helped work on this. And you might say, okay, but does your predicted strategy work? We are waiting to find out with bated breath. So this is just a picture showing that validation experiments are underway that are going to test out these spaced out doses to make sure that what the model predicted is actually working. Okay, I may have gone over, I apologize, but I do want to leave time for questions.
Okay, John?
Oh, thank you, Dr. Jackson. So one question that I had is, once you get these results back, if they do match up with the model, is your next step going to be getting this out drug immediately and then start working on different types of cancer? Are you able to like model in a similar way to the way you modeled this one, or are the models like drastically different?
It's a very good question. So once the validation comes back and so you see this picture has mice in it. So that's the first step, is to make sure it's going to work in mice. The next step after that is to make sure these spacing out treatments work in humans. So there'll have to be clinical trials with humans, testing out these targeted strategies. And eventually, what I would love to see happen is some of these dose regimens actually making it to clinical practice. That would be the best case scenario here. And for me, my next modeling projects all can build off of these. So this project had so many building blocks that I can take with me and use when I want to study breast cancer, or when I want to study lung cancer, or when I want to study bladder cancer, or any type of cancer. There's so many things that are generalizable in this construct that I can take with me. There are things that have to change though, like the specific parameters have to change, but those core structures that were in this model can carry with me.
Okay, very good. So we want to open it up for others to ask questions. you could unmute your mic, your sound. I have a question too. So how long does it take, or did it take? Because it sounds like once, as you just explained, once you have the model in place, you can now just, suit it, fit it for other scenarios. But how long did it take to build this model? What type, did you use a laptop computer or did you use a supercomputer? What, give us a little bit of that.
That's a great question.
Yeah.
Yeah, so that's a great question. So modeling is a process. So I wish I would say that this took a week or a month. This took a long time because when you want to say something about therapies that may eventually impact humans, you want to make sure you've got it right. So we did a lot of calibrating and validating, a lot of pre-processing work, if you will, to make sure our models were predicting the right things and were able to be validated. And that's what took the longest. So I'd say this was probably a couple of years of work, a couple solid years of work to get the model exactly where we wanted it before we started making all these predictions and things like that. Making those predictions didn't take long at all. And we can use a laptop computer to run these simulations. It is we use MATLAB mostly. We don't have to go to our high performance computing for this particular setting. But yeah, so I had undergraduates and graduate students and medical oncologists all a part of the team that helped with this.
Okay. Any other questions you guys have?
I don't have a question, but I'm just, I think that question you just asked, Dr. Murray, is so pertinent. I did a, what do you call it, an MRI recently. And when I went to the place, they told me that they couldn't do it because they didn't have a computer. I was like, wow.
Oh.
So you realize that everything, the calibration and the entire operation was from this technical context of how you have to put in the figures. And so I'm sorry I had a session earlier, so but I realized that when I saw the advertisement that really all these systems of all these machines are mathematically oriented and designed. Right. And if the calibration is off, you could damage somebody, because I was hearing somebody talk about overexposure to radiation or overexposure to something, and you recognize that it's all about the maths, the 0.000001, the 0.5 that really can create. And so it was very awakening for me when they said about the computer, and then I realized that. And then today, when I saw the advertisement, I said, wow, this is something I should be here. I'm sorry I missed it.
Could you tell us, are you calling from Trinidad, USC? Give us a little bit more.
Yes, I am a adjunct lecturer at the University of Southern Caribbean.
Oh, thank you for joining.
What's your field, you said?
I'm psychology.
Psychology. Oh, okay, good.
Yeah. I'm in the Department of Social Sciences. Yes.
Ah,
so I teach grad school and grad and undergrad. Yeah.
Okay, good. All right.
But my area of research is gerontology.
I see. Okay. All right.
That's where all my writings are. Every time I think about it, I have never researched anything in psych. It's always gerontology, the aging. So so most of my publications are in
that
aging.
area. Okay, very good. So we truly have an interdisciplinary audience here, Professor Jackson.
That's wonderful.
Yes. So any other questions or comments?
And thanks for the illumination, Ms. Dr. Jackson, Professor Jackson.
Yes. Yes.
Do you send the recording back usually?
We will get you.
Okay.
You will get you the recording for sure.
I will pay attention.
Okay.
So
another question I have is opportunities for summer research with you or your collaborators this summer.
Yeah, I think there are, so the University of Michigan math department offers an REU, and you can apply for that REU and select me as the person that you'd like to work with. And I would be happy to have some help, some some
Okay.
people interested in learning this interdisciplinary field. I mean, I'm always excited about students who want to use their quantitative skills, even if you're not a math major, just use your quantitative side of your brain to help with these kinds of problems. So yeah.
Okay. I had a sort of interesting, I think, I don't know, it's interesting or dumb question. Where you obviously are in a place where you use both knowledge of math and knowledge of biology. Is there a preferred way to get to that place? Do you start off as a biologist and then learn the math? Do you do the math and then learn the biology? Does it really matter?
That's a great question. And I think you'll get a different answer from a lot of different people. And I might be a little bit biased.
Yes.
But so my answer used to be learn math first and then learn biology second, because that's the way I kind of had to do it. But I don't think students have to do it that way anymore. There are so many interdisciplinary courses out there now. You can almost get a dual training starting at your undergraduate, taking courses together, seeing connections so much earlier than I was able to. So I'm a fan of dual training, and if you can kind of work it out to kind of learn them in parallel.
Yeah.
Why one then the other? Yeah.
Right. Very good. Very good.
Yeah.
Yes.
We have a biomedical degree,
Yeah.
undergrad degree
USC.
at the USC.
Yeah, that's wonderful. Yeah.
Is there, I'm assuming for that degree, there is a math requirement. Maybe they have to.
Yes, there is. Yeah, I could. My granddaughter is doing it, and there
Yeah.
was a math. She didn't get a good grade at the high school level,
Okay.
so she had to do some. She had to do an exam, just entrance. Yes, so they needed her to see if her math was higher than the grade she got. I think if you what we have is 1, 2, 3, 4, 5. I think she got a 3. So because she got a 3, they wanted to make sure that she was a little more proficient. So they had to do the exam. So you're right. There was a math component there.
Okay. All right.
I do see one question in the chat from a student.
Okay.
Ian N. It says, what has to change in the model wise to focus on a cancer in different areas or different intensity of cancer? So that's a really good question. So if you remember back when we were developing that simple model together, we talked about parameters like the death rate of the cancer stem cells. And we said there was the growth rate of the cancer stem cells and a probability that division would be symmetric or asymmetric. All of those are variable across cancer types. So if I were to move to a different cancer setting, I would have different numbers put in for the situation, a situation specific parameter values. So those are the biggest things that need to change as I move from one cancer type to another cancer type.
Okay. Another question for me is, we have a collaborative research going on in 2 areas in biology here. One is collaboration dealing with glioblastoma.
Oh, okay.
It's the one that, a number of famous politicians, Kennedy, John McCain, Biden's son, suffered from.
Yes.
Have you been doing anything or been approached to collaborate with others in that area?
So I've done just a little bit with glioblastoma. There we were looking at oncolytic viral therapies. So we were looking at the way in which so that's such a difficult cancer because it's in the
Yes.
brain. There's the
Yep.
blood-brain barrier, so chemotherapies and things don't work.
Mhm.
So there's a real hope that these viral therapies can work. So we've done some modeling of viral therapies for glioblastoma, but I've only dabbled in that just a little bit. That is such a challenging
It is.
tumor to combat. It really, really is.
Yeah. Other questions? I think one math professor saying that she wants the video so she could share it with her differential equation class.
Yeah, that's great. I actually use this as a teaching model myself. If anyone wants to send me an email, I'll be happy to send lecture notes based on the beginning slides, the equations. Yeah.
Okay, very good. Outside of cancers, have you thought about dabbled in other types? Because the other research that I'm collaborating on right now with biology is bipolar disorder. And it seems to me, based on what I'm hearing from you, that, and knowing the sort of cyclical aspect to the bipolar, 1 minute you're manic, the next you're depressed, and there could be associated some sort of chemical imbalance, in each. That might be a very interesting system to look at, because there's a lot of stuff that's not figured out and known in that area. And some of the current medications are toxic, lead to suicide, and yeah.
Yeah. I think that is a area that's ripe for mathematical modeling. As you said, some of the aspects of bipolar really lend itself to temporal dynamics, like modeling the temporal dynamics of what's going on at the cellular level. So yeah, I definitely think that that area could be very fruitful in math.
So maybe we could talk offline?
Yeah, sounds good.
Okay.
Okay.
All right. Anybody else has a question, comment?
Oh, I just had one quick other question. So I was wondering, so if someone who has a background in both math and biology, do you think that they are better to make a math model than someone who has a background in biology and a little bit in math, and someone who has a background in math and a little bit in biology?
Oh my, that's a tough question. So we're moving into such an area of team-based science where you bring whatever expertise you have to the table and it's going to be welcome and utilized. So I think all three of those types of students have the potential to build equally good models. I definitely think that knowing the underlying biology helps so much, because you know that you're not going to be able to model the entire cellular, molecular, genetic landscape that's going on. So that background will let you know what realistic simplifications can I make. And someone from a math perspective would probably just cut out all the important stuff, right? So, I mean, I think there's a balance. I think each three of those types of students would be able to work, especially if they could work together, would come up with amazing models.
Have you worked with chemists before too?
Say again, I'm sorry.
Have you worked with chemists as well? Because I noticed, of course, you made the plug for the math modelers, but chemistry does contribute to cancer.
You are absolutely right. And I should have made a plug for chemistry too, because actually my very first collaborators were biochemists from Bristol Myers Squibb Pharmaceutical Company, who were developing these new drug targets. So I don't know how I forgot that, but yeah, you're absolutely right. So that was my first real collaboration, happened to be with biochemists.
Biochemists, yeah. Okay. All right. If there are no more questions, we definitely want to thank you so very much for this. I see John. Is that?
Oh yeah, that's just one other question I was wondering about. Sorry,
Okay, we can take that as the last one.
Oh, okay. So when you make this model, can it be like tailored specific to a person, or once you get to optimal treatment plans, is it more just you run those in general?
That's a great question, John. You've asked such pointed questions that are right on target. You know, that is where this field is moving. I can't tell you that we are there yet, but patient-specific precision medicine is where we're going. That's the biggest, most powerful use of these kinds of mathematical models. We're not able to take exactly information from a specific person's physiology, chemical makeup, genetic makeup, and put it into our model yet, but that's the direction we are moving into.
That's very excited for personal medicine.
Yeah.
Very cool.
John, as a psychologist, that's my heartache, because I think we are affecting emotion, which will affect the body chemistry, the limbic system, etc., which will affect the outcome.
Yeah.
And I think we really need to be collaborative in working together, with psychology, chemists, everybody, to get that specific, because each individual responds differently.
Absolutely.
So just a touch, just the thought of hearing, I have to do a procedure. I think it's something we have to work towards. And,
I absolutely agree.
it's one of my
100%
peeves. Yes.
Yeah.
And well, this is a good segue, because actually next week, the title is The Future of Precision Medicine.
Perfect.
That's next week's presentation.
I
So
might join you for next week's too.
Oh, come on. Yes, you, your students, whoever. Yes.
Okay.
All right. So again, we want to thank you so very much. Great talk, very inspirational. And we hope you all the best in the future. I will definitely be keeping in touch with you about
Wonderful. It's been my pleasure.
Yes. Thank you so much.
Okay. Thanks, everybody.
Okay. Take care. Bye-bye.